So Fraser gave a really nice talk about what you can get out of FFPE, and also he touched upon at the very end the technical problems that FFPE samples really present us with. And as a scientist by training, so I'm not a pathologist, I'm not a medic, I'm innately skeptical. And when I embarked on the project that I'm going to tell you about, I was skeptical from the outset. So I wanted to truly test the technology to see whether or not we could actually get anything meaningful out of FFPE samples. So we've heard before that FFPE is degraded. This is a really nice bioanalyzer plot here showing undegraded cell line RNA with 2 ribosomal peaks. This is a representative cervix sample that's been stored for about 10 years. And what you can see is instead of these peaks, you end up with a bump of what is essentially degraded, cross-linked, mutated RNA. We know that DNA is a lot easier to work with and protein even more easy, but FFPE RNA is probably the worst thing that you can come across in terms of study. And that's, as Fraser alluded to, because you're looking at quantitation rather than absolutes or digital differences. So we know that there are so many samples out there that it would be horrendous to ignore them. So really what we want to see is how far we can go back, what information we can get out of samples. So the work that I started, when the kind of the current state of affairs when I started this project was some work that had been done previously at my centre, showing that if you go from standard Affymetrix labeling using U133 arrays, which are the same arrays that Fraser had used, and then use what's called NuGen, which is a method of amplification of RNA, the concordance between fresh frozen and FFPE jumps from about 50% to about 80%. And then if instead of using the U133 arrays, you use Exon arrays, which are an array type that have far more probe sets, essentially targeting all of the exons in the genome, you can see that that concordance increases a little bit further. So this kind of gave us hope that it wasn't just all noise in these samples, but what we really wanted to know was, could we derive a biological signature, something that was intelligible, without having to have paired samples? So Fraser has fantastic paired samples which he can interrogate in a lot of detail, but the problem is that these millions and billions of samples that are out there don't have these paired samples. So we wanted to know, what could we get out of these samples. So this is essentially a schematic showing how the exon arrays differ to 3' IVT arrays like the U133, and essentially it is just more probe sets, so you have more depth throughout the genome. The HTA arrays, which Fraser touched upon, are reversion beyond the exon array, and it's something that we're also looking into as a method for FFPE analysis now. But essentially what we do is we take the FFPE sample, we extract the RNA, treat with DNAs, do cDNA synthesis using NuGen. We've also done Sensation Plus, which is Affymetrix' new amplification method, and we find that to be equally good as NuGen. We essentially label and put onto the arrays. So the problem is when you have samples that you don't know what the quality is going to be like, you want to have something that's black and white, something that you know is going to be different. And if there are any pathologists here, which hopefully there are, one of the big differences in cervical cancer is the difference between squamous cell carcinoma, which is P63 positive, and adenocarcinoma, which is negative. So we use this design using clinical samples which had clinical associated data that were taken from our standard pathology stores at the Christie Hospital. And we just took a handful of arrays just to pilot the idea to see whether we could detect differences between the squamous cell carcinoma and the adenocarcinoma. So one disclaimer to this is that obviously this is a rather large difference. In terms of morphology, in terms of gene expression, these are really big differences. And the reason that we're looking for really big differences is because we have really small numbers. If you're wanting to look for really small differences like survival, you have to increase the numbers. But these numbers, this design allows us to address the question that we wanted in terms of feasibility. On the bottom of the slide here, we have DABG, which is essentially a metric of how well the arrays work, so how much signal there is. And on this axis here, we have age of sample. And what you can see is that most of the arrays, so most of these 28 arrays, have detectable levels of information in them. But that doesn't mean that that information is usable or intelligible. It could just be noise, like Fraser showed in the previous RNA-seq example. So what we did was we took the samples, and without doing anything to them really, except for looking at them, we asked how well do they cluster. And the very first thing that we see is that they separate into adeno and squamous quite beautifully. So this is looking at the 1,000 probe sets with the highest variation in the experiment. But essentially the same thing is true if you look at all the probe sets on the array. And this here is a PCA which is very similar to the MDS plot that Fraser showed. And what you can see again here is just, it's another way of visualizing the separation between the samples. So what that shows is that the main difference between the samples is not technical variation, it's biological variation. So that gave us some hope that what we were seeing was not just noise. So we then went through a bioinformatics pipeline, and this is a very standard bioinformatics pipeline for exon array analysis, and it's kind of referenced kind of ad infinitum. But what we end up with is a list of probe sets and genes which are different between squamous cell carcinoma and adenocarcinoma, so things that are up in one and down in the other. So this slide really is kind of the conclusion of the proof of principle experiment. So essentially what we wanted to see was, could we see a difference between adeno and squamous on the arrays? So these samples were on average about 13 years old, so they're proper bona fide FFPE samples. And what you can see here is a cluster dendrogram. Each one of these rows represents a different probe on the exon array. So if you imagine this is the gene here for p63, all of these different probes along the gene length. And what you can see is that red is detection of the RNA and blue is low levels of detection. So you can see that you're not just getting a single measurement of whether or not there's a difference between adeno and squamous, but you're actually getting 18 independent measurements. So this is one of the advantages of using the exon arrays or using the HTA arrays. It does make the bioinformatics slightly more complicated, but it's we think it's a much more powerful tool. But okay, we derived a signature in FFPE. It could still just be noise, even though everything looks hunky-dory. We wanted to make sure that it was truly a signature that could validate. So obviously we needed to go to an independent data set. And at the time when we looked for a cervix cancer data set that we could use for adeno and squamous, there weren't any, so we ended up looking at lung cancer. And at the time I was quite skeptical that it would work in lung cancer because I thought the cervix and the lung were quite different. But surprisingly, it actually does separate quite nicely into squamous and adeno. And again, this is the PCA plot showing the difference. You can see here really in the cluster dendrogram where there are genes which are associated with one of the histotypes and not with the other. So what this shows is that if you take the genes that we've identified from bona fide FFPE samples, trained on a signature, on the exon arrays, and then migrate to fresh frozen samples, which are on so these are on the U133 arrays as well, so it's a different array type, you still see consistent biology. So this really validated the signature that we found. So once we generated the signature, we wanted to move forward with that, and one thing is that most people then tend to do is to try and validate by QRT-PCR or some other kind of quantitative method. I unfortunately had done a lot of qPCR in my PhD and I wasn't going back. It's a horrible technique, particularly when you're looking at hundreds of genes. So we came across Quantigene, which was Quantigene Plex, which is a technique that instead of requiring RNA isolation, reverse transcription, and then the actual test itself, essentially you take your sample, you can take a direct lysate from the FFPE, mix with some beads which are impregnated with probe, hybridize overnight, and then get results on what is a glorified flow cytometer. So we thought that this might be a very good way and a very quick way to validate a lot of our array data without the huge amount of hands-on time that would have been required for the QRT-PCR. So the way that Quantigene Plex works is that you have beads. Each bead has a slightly different colour. It's a mixture of two fluorophores. There are various different components to the Quantigene which bind specific RNA to the beads and only the RNA from the gene of interest. But the clever thing is the branch DNA technology, which makes it so sensitive. So instead of amplifying like we do with PCR, we essentially build a tree-like structure, hence branch DNA, and it's this which is then visualized as fluorescence in a Luminex machine. So this is almost like immunohistochemistry at this point. So it's kind of, we thought that this made sense to work with FFPE samples because immunohistochemistry works quite well as well. So we designed probe sets that overlapped with the differentially expressed probe sets that we had on the Exon array, and we compared, and then we compared to the array data, and this is what we got from the Quantigene. So basically, again, what you can see is that the genes that we'd taken, so we took the top genes from the adeno and the top genes from the squamous, and you can see that a large number of them, pretty much all of them with the exception of one, validate really, really nicely. And again, you can see the separation into the two different histological types. So this shows that this platform actually works quite well. The real magic of this is that all of this data was generated in essentially, I would say, a 48-hour period. So essentially, you hybridize overnight, but the amount of hands-on time with this assay is so quick that it is actually like magic. And for someone that's done a lot of QRT-PCR in the past, it's a hell of a lot easier. And the data is far more robust. We've done other studies where we've compared directly to QRT-PCR, and the correlations with array data is much higher with Quantigene. So it does seem to be a very nice method. These are the correlation coefficients for Quantigene to the array data, so you can see that a large number of them are above 0.5. So we postulated that, well, that's everything done. There's no longer a problem with FFPE. We can go back and take samples for 25, 50 years. And we hybridized samples. So we went back, so we took a cervix cancer cohort and a bladder cancer cohort. And the first thing that we asked was there anything that we could learn about these cohorts in terms of how well they performed? So looking at percent DABG, which I told you previously was this measure of how much biology had been captured by the array. So the first thing that we found was that RIN, which is a really, really popular RNA metric for good quality RNA, actually has no correlation with how well the array works at all. So it's no correlation at all. We see that in both data sets. So we suggest that with FFPE that RIN actually shouldn't be a reason not to hybridize the samples, which when I started in the group I was told that a RIN less than 7, you shouldn't hybridize the sample. All of my samples had a RIN less than 7, so I wouldn't have hybridized any of the samples. So moving on from that, what we did see, and this is what Fraser alluded to, is the fact that as samples get older, so if you look here, the age of the samples, and this is a separate publication looking at QRT-PCR, that essentially as samples get older, their DABG decreases. So these samples here are about 10 years compared to these ones here, which is around 20. And what we wanted to know was this decrease associated with a loss of biology? And unfortunately it was. So what you see here is the training samples that I told you about, the adenosquamous, and this here is a ratio. So adenosquamous. But when you look at the older samples compared to younger samples, so there was a bifurcation in the cohort. These were two different trials for two different purposes. So if you split them, you can see that the younger samples have biology. They can tell you whether they're adeno or squamous, but actually the older samples lose that biology. And that biological loss is progressive. So as samples get older, the ability to tell you whether a sample's adeno or squamous decreases. So you can see that here with P63. So there was a lot of talk at the time about microRNAs and how stable they were in FFPE. So we postulated that maybe in these samples that were so old, so things that were 20 years old, that the mRNA might have degraded to the point at which they can't be measured, even using Exon arrays, which have these tiny little probe sets in them that should be able to pick up the degradation. So we then looked at microRNAs. So we were quite lucky that P63 has a microRNA that correlates highly with it. So this has been described previously in lung cancer. It's a microRNA called 205. And so this is just showing in the array data the correlation of the mature probe set for 205 along with the TP63. So we then asked the question, can we see a difference in the older samples? So here we've got cell lines, young samples, and old samples divided into groups. And what you can see is there's a nice big difference in your cell lines, but as the quality of the RNA decreases, the difference between the samples decrease as well. And that's true for both of the microRNA, both of the mature microRNA probe sets that are on the exon array. But what's not on the exon array are the processed microRNAs. So when we talk about microRNAs, we talk about these very small processed forms. So on the array is the pre-microRNA, the exon array. So we then thought, well, let's throw good money after bad and buy some more arrays and see whether or not we can actually see a difference in these samples that we couldn't see a difference. So we started off, though, before we looked at the arrays, by doing qPCR using TACMan. And what you can see is in the older samples, so this is now looking at the mature form of 205, that there's a nice big difference between the adeno and the squamous samples. So this is the difference that we couldn't see before with the mature form on the exon array or with P63 on the exon array. So then we did the same thing on arrays, and this again here is the array data just for the subset of samples. So these graphs at the bottom look horribly complicated, but actually they're quite simple. On this axis we have young samples, this axis we have old samples. And what you can see here is a bump, and this is from the exon arrays, and that bump represents a shift in signal representing higher expression in the young samples, whereas the older samples have less expression. But when you look at the microRNA data, so anything that's expressed is quite linear. There's no deviation, there's no bump like we see with the exon array data. And that's true also for SNO RNAs and SCAR RNAs, which are on the array as well. So what we did was we decided to lump our samples together, our two groups, the old and the young, which gave us, I think it was 8 squamous and 8 adeno. And we asked the question, could we see a difference between the samples? And then microRNA 205 was statistically different. So what this shows is that microRNAs can actually rescue samples where mRNA is degraded in them. And another way of showing that is this graph here. So these are just box plots representing adenocarcinoma and squamous carcinoma. So this here is P63. This here is the microRNA probe set 1 and 2 on the exon array. And this here is the microRNA data for 27 matched samples. So you can see that there's no difference unless you look at the microRNA expression. So this is where we were at maybe about a year ago. So we kind of now have this almost decision tree. So essentially, there's very little that you can do up front to QC an FFPE RNA sample. We don't really know what it is that makes the samples work and what it is that makes them fail. But what we do know is younger samples have a much higher likelihood of working. Younger samples, you can do exon array profiling, U133 profiling, Quantigene Plex, and that all works wonderfully. But as samples degrade beyond that, you lose the signal in mRNA. But that doesn't mean that the samples are worthless because the microRNA profiling is still an option. And again, this is a lot of work supported by a lot of different people and departments. So I'd like to thank them all. Thank you.